Welcome everybody to a StriaTech hosted journal club. My name is Thomas Münch and I'm one of the founders of StriaTech. I welcome you to another of our journal clubs where scientists present their projects and data, highlighting the use and the application of our products. StriaTech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The StriaTech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Supraja Varadarajan as our speaker. Her talk is entitled Postsynaptic Neuronal Activity Promotes Retinal Axon Regeneration. Supraja came from India to obtain her master's degree in biomedical engineering at the New Jersey Institute of Technology. Afterwards, she worked for 2 years at UC Irvine as a staff research associate before starting her PhD in neuroscience at UCLA in 2010. From 2017 until now, she has been working with Andrew Huberman at Stanford University, where she has done the study that she will present today. From January of next year, she will head her own lab at UT Southwestern Medical Center. We are happy to have you as a speaker today, Supraja. Before you start, let me remind the audience that you will be able to ask questions throughout the event, and also to give a thumbs up to other questions. We will get to those questions at the end of Supraja's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, and you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will stay available for a couple of days after the live event today. So if you're currently not watching live, but a recording of the event, and if Q&A is still available, please make sure to enter your question and your name so that we can back to you with the answer. We are very much looking forward to your questions. So Supraja, please go ahead with your presentation. Make sure to unmute your microphone so that we can hear you and share your slides. Thank you.
Thank you, Thomas. Thank you for the warm welcome. And I'm very excited to be here. And I want to start by thanking Thomas and Lena and the others at StriaTech for this wonderful opportunity. Let me quickly share. Okay, can you see my slides okay?
Yes, it's good.
Okay. Perfect. So I'm very excited to be sharing this work that was recently published in Cell Reports as of last month on how postsynaptic neuronal activity promotes retinal axon regeneration. These postsynaptic neurons that I'll be talking about reside right here in the subcortical region in the mouse visual pathway, in the mouse adult brain. And these neurons receive visual input. And today I'll be talking to you, I'll give you a brief background that's relevant to my work, and then I'll tell you about the distal injury model that we designed to specifically address this question, and then talk about how increasing activity in these neurons promotes axon regeneration of retinal ganglion cells. So vision, as we all know and use it, is a critical sense that humans use to survive. But today, what I want to remind us is that we use vision for a variety of functions, such as image-forming functions, such as identifying objects and navigating the space around us. But we also use vision for a variety of non-image forming functions, such as sensing the light around us, which sets our circadian rhythms, and for image stabilization so that we can make sense of objects as we move around in space. Otherwise, everything would be a giant blur. But when a disease or an injury affects the visual system, it severely impacts the day-to-day quality of life for those patients. And here is an example of how a patient suffering from glaucoma would view their world. Now, glaucoma is one of the leading causes of blindness, which affects about 70 million people worldwide. And patients start by losing their peripheral vision and slowly lose their central vision as well. And apart from glaucoma, there are many other conditions that can also lead to blindness, such as eye injuries and optic neuropathies. So, needless to say, that there is an immediate need to identify strategies that can restore vision. Now, if you look at the mouse visual system, here's a little schematic to orient you. You have the eye, which houses many types of neurons, but I'll focus on the retinal ganglion cells, or the RGCs, which are the only neurons within the eye that extend axons out of the eye and relay visual input to these postsynaptic central targets in the brain. Now, there are many different subtypes of retinal ganglion cells, but each of those subtypes are responsible for encoding visual information about distinct visual aspects and relaying that onto distinct visual target nuclei in the brain. For example, a subtype of RGCs receive information, encode information about circadian rhythm or brightness, and relay that onto the suprachiasmatic nucleus. There's information encoded about motion detection, relayed onto the lateral geniculate nucleus. Information about pupil reflex and image stabilization relate to distinct centers in the pretectum, which is the region I'll focus on. And finally, visual information is related to the superior colliculus, where it's integrated with other sensory information. So it's a very highly complex pathway, and it's formed in a very directed and stereotypic fashion. So let's take a quick look at how these visual circuits are formed with so much precision. Now, I really love this diagram because it's a beautiful diagram from none other than Ramon Cajal. And to me, the beauty of the system lies in its complexity. And in the way in which these RGCs find their axons find their way out of the eye, navigate their path to the brain, find their targets, and form synapses with those postsynaptic target neurons. And the precision with which these presynaptic axons interact with postsynaptic targets is ultimately what produces vision. And you can appreciate that complexity here in this more modern day representation, modeled after the London Tube, where these are approximately 45 RGC subtypes that as a population map onto approximately 40 visual target nuclei. So again, it's a highly directed process where each of these subtypes take a stereotyped pathway to identify and connect with their postsynaptic targets. Now, let's just take a quick look at how these circuits are actually formed. We know that during development, these RGCs receive guidance cues from within the retina, using which they extend their axons out of the eye and down the optic nerve. They then cross at the optic chiasm and extend into the brain, where they form synapses with these postsynaptic targets. Now, what's interesting is that as these axons extend closer to those postsynaptic targets, they also receive guidance cues and trophic factors from within those postsynaptic targets. Neural activity becomes really important to refine those axonal arbors within those targets. But the main point that I want to highlight here is that as these RGC axons extend and form visual circuits during development, they receive signals from both within the retina as well as from these postsynaptic targets in the brain. So when a disease or an injury affects the visual system, these axons disconnect from those targets, the axons degenerate, the cells die, and it leads to vision loss. And so we can talk about this for a whole day, but I'll just generalize a little bit to summarize that in general, our regenerative capacity reduces with evolution and age. And so if you take humans who are, especially adult humans, who are the most highly evolved species, they have very little regenerative ability, especially in the central nervous system. And because these RGCs are part of the central nervous system, they retain very limited regenerative potential. So, and there's many different reasons that lead to this regenerative failure, and they can be broadly classified as intrinsic factors that are intrinsic to the RGCs, such as lack of neurogenesis. So there are no more new neurons being born throughout their life, as takes place in fish, for example. There's a downregulation of growth promoters and an upregulation of growth suppressors. And then there's extrinsic factors such as the glial scar, myelin-associated inhibition, and prolonged inflammation. And some of these are actually beneficial responses, but prolonged responses become inhibitory to regeneration. So if we want to restore vision, then we need to achieve at least three major milestones. We need these cells to survive, or we need to replace lost cells. And we need those surviving cells to then regenerate their axons and find their correct paths. And we need those regenerating axons to reconnect with their targets to restore vision. Now, a very successful strategy in the field has been to reapply fundamental principles that form visual circuits during development towards regenerating those same circuits following injury or disease. And a really good example of this is a landmark study done in Shigangi's lab using the optic nerve crush model. Now, they showed in 2008 that if you crush the optic nerve behind the eye in mice, these axons degenerate to the point of lesion. So the lesion site is shown by the asterisk, the retina is on the left and the brain is on the right, and these axons die back to the point of lesion. But they showed that if you knock down P10, which is an intrinsic factor, then you can promote regeneration of these adult CNS axons, which was a huge feat and has since become the sort of the gold standard for regeneration. But since then, there's been many groups that have done some phenomenal work to identify transcription factors, growth promoters, groups of transplanted RGCs, and our lab has previously increased neural activity in RGCs. And together, these groups have achieved huge strides in our understanding of how to promote cell survival and promote axon regeneration. But there's still a lot left to understand how to get these regenerating axons to then reconnect with their postsynaptic targets. And moreover, all the approaches that we know of thus far have used presynaptic perturbation. So they've directly manipulated mechanisms in the RGCs, their axons, or their presynaptic inputs. And so that got me wondering about postsynaptic cells. And so when I started my postdoctoral work, I was wondering what happens to these postsynaptic cells after injury. Now, if you look at the visual system, this is the case for many other systems during development generally, where the target neurons also provide essential signals to guide axons toward those targets. The same is the case for the visual system. During development, I'd mentioned that as these axons reach their postsynaptic targets, they receive guidance cues and trophic factors from within these targets. Neural activity is also important to refine their axonal arbors. And groups have shown that if you lesion these postsynaptic targets or deplete target-derived signals, then that increases cell death and leads to incorrect wiring during development. Suffice to say that, again, that there are important signals coming from both within the retina as well as these postsynaptic neurons during development that are crucial to form these circuits. And yet we understand very little about what's happening with these postsynaptic neurons after injury. And so one of the first things that I wanted to address was what happens to these postsynaptic neurons, and is there a way to leverage these neurons to promote RGC axon regeneration. And the first thing that I did was to use a distal axon injury model that I'll explain shortly. Now, I've talked about the optic nerve crush model, where you basically take a forceps and you crush the optic nerve behind the eye. Now, this has been sort of the common model to induce an optic nerve injury and induce degeneration. And it's a very severe injury, which causes massive cell death, such that 80% of the RGCs are dead within 2 weeks. And these axons degenerate to the point of lesion. So there isn't very much left to work with at this time point after a week or 2 weeks, because the cells are dead, the axons have degenerated almost up till the eye. And so at this point, we're not even at the point where we can begin to address what the postsynaptic cells are doing. Now, in the distal axon injury model, I'll first go over the basic technique that you're probably familiar with. We can inject an anterograde tracer, such as toxin B, conjugated to a fluorophore into the eye. And this is really good at labeling the RGCs and their axons, as you can visualize in green. These axons ascend as the optic tract in the brain, where they extend collaterals into two major targets, such as the lateral geniculate nucleus and the superior colliculus. And they also extend collaterals into many smaller targets in the pretectum, shown by these white dotted circles. Now, in the distal axon injury model, I severed the optic tract in the brain anterior to the pretectum. And there's many reasons to use this injury model as opposed to the optic nerve crush. One is that because these axons innervate many other visual targets anterior to the site of lesion, they presumably retain trophic support from those targets. And there's now a much shorter distance over which these axons need to regenerate back to the target site. And this short distance would hopefully allow us to closely monitor targeting choices or errors. Now, the first thing I did was to assess if this injury meets our criteria. And so I looked at cell death in the retina. So I labeled these RGCs. I collected whole mount retinas from two groups. One is a sham group that doesn't undergo injury, and the other is a control group that undergoes a distal injury. And 2 weeks later, I collected retinas and stained them for RBPMS, which is a marker of RGCs. And I found no significant difference in the number of cells, either in the ipsilateral or contralateral eyes or between the sham and control groups. So at a 2 week time point, RGC cell survival remains unaffected by the distal injury. So that was one criteria that was met. I next used CTB labeling to look at axonal innervation in some of these anterior targets that I mentioned. So here you can see RGC axons ascending in the optic tract. They innervate the SPN, the suprachiasmatic nucleus, one of the earliest targets in the brain, and you can see enough innervation that's retained after 2 weeks of distal injury. And the same with the ventrolateral geniculate nucleus, and one of the big targets for most RGCs anterior to the lesion. So they retain connections and would hopefully continue receiving trophic support from these targets. So next, I wanted to understand if these RGC axons somehow spontaneously regenerate following injury. And I think we all know the answer to this because these are CNS axons. There isn't much hope. But nevertheless, to evaluate it, I used a dual tracer approach. So I injected a pre-injury tracer into the eye to label the intact visual pathway as seen in green. And then 2 weeks later, I injected a post-injury tracer in red, which is also CTB, but conjugated to a different fluorophore. And I used these two tracers to identify regenerating axons. Now, I'll just take a moment here to emphasize that this is a partial injury model. So there are spared axons that remain uninjured. And this is more clinically relevant to some injuries that we see, or even in disease models where some axons are affected and they degenerate, but there are axons that remain connected and survive just fine. And so these paired axons in the distal injury model would take up both the pre-injury and post-injury tracer and appear yellow. And I can basically use these tracers to identify each of these individual components or any regenerating axons, which would only be labeled by the post-injury tracer. I'll show you what I mean by that. Here is a sagittal section of a mouse brain labeled with a pre-injury tracer. The white dotted line indicates the lesion site, and these are CTB labeled RGC axons innervating pretectum targets. Now I can threshold these image and receive a result as shown on the right. Here is my post-injury tracer. And I can run these images through a specific algorithm to pull out the red-only pixels, which would indicate that they are regenerating axons. So I can unambiguously identify regenerating axons. Now, as we would expect, because these are CNS axons, without any treatment, there's very little regeneration going on, as you can see. So how do we promote regeneration? Now, neural activity has been shown by many people and largely by Mike Greenberg's work that neural activity induces gene expression changes, and that these gene expression changes can manifest both as synaptic changes as well as morphological changes. Our lab, Jeff Goldberg's lab, Jeff Goldberg, when he was in Ben Barris's lab, has shown that both electrical activity and trophic factor signaling promote RGC axon extension during development. And a previous graduate student in the Huberman lab has shown that when you combine neural activity in the form of visual stimulation with an intrinsic growth factor, such as increasing mTOR, you can promote RGC axon regeneration to long distances. So safe to say that neural activity was a very attractive candidate by which to promote regeneration. And so what I did was I used the distal axon injury model, and I wanted to test the hypothesis that increasing activity in these postsynaptic targets can also promote regeneration of RGC axons. And so I used a chemogenetic approach to increase activity in these pretectal neurons. And I then performed a distal injury anterior to the lesion site. I then used my dual tracer approach to ask if increasing activity distal to the lesion site can promote regeneration of these RGC axons. Now, just a quick reminder for those of you who are not familiar with chemogenetics, here I'm using a designer receptor exclusively activated by designer drugs or DREDs. And so in this case, I'm using an AAV to express an HM3D DRED, which is a modified human muscarinic receptor. And so this HM3D is modified, so it can only be activated by an external synthetic ligand, such as clozapine N oxide or CNO. So when the CNO binds to HM3D, it increases firing or neuronal activity in those infected neurons. I collaborated with Fei Wang and Jin Duan's lab at UCSF, and Fei very kindly recorded from these HM3D expressing neurons to confirm HM3D works the way it does. And he confirmed that when you activate, when CNO binds to these HM3D expressing neurons, you have increased firing. I also confirmed that these HM3D neurons are expressing CFOS, which is a marker for early gene expression induced by activity. And so you can see here that in controls, there's very little CFOS expression because these mice have been carefully dark adapted and taken precautions. But when you increase HM3D when you increase activity for 24 hours, you see a significant increase in expression in CFOS in these HM3D expressing neurons. And this increase in CFOS is sustained for even over a 2-week period of increasing activity. So I injected an AAV in the pretectum so that these blue neurons shown over here would express HM3D and be activated by CNO and have increased activity. And then 2 weeks later, I injected CTB into the eye with the green tray of fluorophore. So this is my pre-injury tracer. And so this marks the entire intact visual pathway. 2 days later, I performed a distal injury to sever the optic tract and then started injecting one cohort of mice with CNO so that those pretectal neurons expressing HM3D would have increased activity. And then 12 days later, I injected CTB conjugated to a red fluorophore, which is my post-injury tracer, to identify regenerating axons. I'll show you a quick video to orient you as to where in the optic pathway the distal injury is, and in relation to that, where the injection site is. So these are CTB-labeled RGC axons ascending as the optic tract, innervating the DLGN, the pretectum targets, and the superior colliculus. And here in blue is where I inject HM3D to express HM3D. And just anterior to that is where I sever the optic tract. So again, the idea is to sever the optic tract and increase activity distal to the lesion site and ask if they can provide any target-derived signals that promote regeneration back to those targets. So I've shown you this example before from a control animal, which only receives a distal injury. And again, because these are CNS axons, without any treatment, you see very little regeneration. But when I increase neural activity in the pretectum, I see a significant response in regeneration. If a quantification of this pixel density shows that compared to controls where there's very little regeneration, there's significantly more regeneration in the activity group. Because this is a partial injury, we also quantified the spared axons and wanted to understand if these spared axons somehow contribute or confound regeneration. But we found no significant difference in sparing between the two groups, which basically indicates that this increase in regeneration is attributed to the increase in activity in those mice. I then wanted to understand over the extent to which increasing activity can promote regeneration. And so if you look at the quantification here, you can see that 0 is where the lesion site is, which is where which is where I sever the optic tract and perform the distal injury. And so anything to the left of that red bar is anterior to the lesion, whereas anything to the right of that bar is posterior to the lesion. And this blue rectangle here represents the region over which I'm increasing activity. So the control mice here in gray have very little regeneration, only up to about 400 microns past the lesion site. But increasing activity significantly boosts regeneration all the way up to 2 millimeters past the lesion site. And so this is sort of comparable to that gold standard that I mentioned before, where they see regeneration up to 5 millimeters in 4 weeks, and we're seeing regeneration up to 2 millimeters in 2 weeks. Now, a good measure of regeneration is how well these regenerating axons reach their targets and restore function. And so those are the next two questions that I asked. And I'll show you all of the individual targets that we've looked at in the pretectum. Now, this is the top view of the optic pathway, and the white dotted line indicates the region over which we increase activity, shown in blue. Now, if you look at the olivary pretectal nucleus, or the OPN, which is one of the pretectum targets that's responsible for controlling pupil reflex, which is how well our pupils dilate and constrict in response to light, we see that compared to controls, we see more regenerating axons innervating the OPN. We looked at the nucleus of the optic tract, or the NOT, which is responsible for horizontal eye movements, and noticed the same increase in regenerating axons innervating the NOT. And the same for the MDPPN, although not much is known about the function of the MDPPN. So these regenerating axons innervate all of these minor targets in the pretectum. Now, we next looked at the major targets, which is the DLGN. And just to remind you, the DLGN is actually anterior to the lesion site. And yet, we noticed significantly more regenerating axons in the LGN. And so we were initially surprised by this, but some beautiful live imaging studies in the spinal cord as well as in the optic nerve have shown that if these regenerating axons, or the injured axons, basically die back about 400 to 500 microns and then begin regenerating. And so this is in line with what we're expecting there. Now, next we looked at the superior colliculus, again, another major target, especially in mice. About more than 90% of RGCs innervate the superior colliculus. And compared to controls, we see significantly more regeneration in the superior colliculus as well. So this tells us that these regenerating axons navigate towards both minor and major targets. Now next, and so this basically tells us that stimulating neurons posterior to the lesion site can promote regeneration of RGC axons. Now, next we looked at whether this regeneration actually leads to rescues any function. And so we used the optomotor response, and we were really, really grateful to be able to use the optodrum. Our collaborators, Jin Duan, the Duan lab at UCSF, had an optodrum, and so we collaborated with them, and this really worked out very well for us at a very opportune moment too. Now, just to go over it briefly, the optodrum is basically you have a pedestal over which you place a freely behaving mouse, and so it's not restrained otherwise. And it's surrounded by these 4 monitors on which the stimulus is displayed. And the software can track the nose, head, and body movements and generate vectors to identify if the mouse can track the moving stimulus. And the moving stimulus in this case are these black and white bars. And what we want to do is, or what the software does very nicely, is to toggle between really low spatial frequency to really high spatial frequency until you can identify the threshold up to which mice can detect that moving stimulus. So we collected 2 responses. One was at day 11. So 3 days before the distal injury, we collected a baseline optomotor response. And the reason is because you can order wild type mice from JAX, and each of them, without any manipulations, each of those mice would have different baseline thresholds. And so to establish a baseline, we collected a pre-injury response. And then approximately 12 days after injury, we collected another optomotor response, which would assay if there's any deficit in function and or if there's any rescue. Now, I'll walk you through this graph here. The gray bars, we have our 2 groups, control and activity. And these light gray bars indicate pre-injury responses, so the baseline responses. And the dark gray bars indicate post-injury responses. And what we see is that in control groups, the distal injury causes a deficit in the threshold that the mice can detect, the threshold of the spatial frequency. But increasing activity allows these mice to retain the same threshold as they had before injury. Now, I like looking at this in a different way, which is if the pre-injury response is 100%, what is the post-injury response? So you normalize the post-injury response to each mouse's own pre-injury response. And so again, what we're seeing is that the distal injury causes a deficit in the threshold that the mice can detect, but increasing activity allows these mice to retain that threshold, which means they can continue detecting the stimulus as well as they did before injury. And we see the same trend in tracking duration, which is how long the mice can track the stimulus. And so we were really surprised and very pleasantly surprised by these results, because this tells us that not only is a partial injury enough to cause a deficit in function, but also that increasing activity can rescue those deficits caused by distal injury. And so now I was very excited because we had this established paradigm with a specific injury to address the role of postsynaptic neurons, and we had a means by which to promote regeneration. So I next wanted to use this paradigm to ask a more specific question, which is to address the role of retinorecipient neurons. And so far what I've shown you is that I'm broadly increasing activity in the pretectum targets. So this includes neurons that receive visual input, RGC input, that is the retinorecipient neurons, but it also includes neurons that do not receive RGC input. So they're just neurons in the vicinity of these retinorecipient neurons. And so to get at the exact contribution of these retinorecipient neurons, I focused on one particular visual target, the nucleus of the optic tract, or the NOT. And to specifically target these retinorecipient neurons within the NOT, I collaborated with a previous postdoc in the lab, Onkar Dande, who screened a bunch of mouse lines and identified the synaptotagmin 17 Cre line as one of the targets in which there are Cre cells within the NOT. And so in this GenSat image, you can see the NOT outlined in gray, and which has Cre cells within the NOT. Now, I first characterized this mouse line better. So by crossing the synaptotagmin 17 Cre mouse line to a TD tomato reporter, you can visualize the Cre cells with TD tomato. And I confirmed that there are Cre cells present within the NOT, but not within any of the other visual targets, like the OPN, which is a pretectum target, and the DLGN, which is a major target for most RGC cells in the mice. And so this was a really nice tool with which to access retinorecipient neurons within one visual target. I then wanted to identify the inputs to these Cre cells. And so I used the rabies tracing system. And just briefly, I injected an AAV helper virus, a Cre-dependent helper virus, to express TBA and the glycoprotein within the NOT. And then 3 weeks later, injected the rabies M-cherry virus into the NOT to label the presynaptic inputs to those Cre-expressing cells within the NOT. And what we found is that you have these yellow starter cells in the NOT, which basically tells you that they express both these viruses. And we found M-cherry labeled axons in the optic tract and traced them to the retina to identify to see M-cherry labeled RGCs within the retina as well. So this tells us that this synaptotagmin 17 Cre mouse line labels bona fide retinorecipient neurons in the NOT. And so those neurons in the NOT receive monosynaptic inputs from RGCs. So I was very excited to be able to use this tool, because now I could use the synaptotagmin 17 Cre mouse line and inject a Cre-dependent HM3D into the NOT. So now only those Cre retinorecipient neurons would express HM3D and have increased activity. And so I followed the same paradigm as before with my dual tracer approach and the distal injury. And here's a little example of that. So these are coronal sections of the mouse brain where the NOT is outlined in white. Now, one of the ways in which we identify visual targets is by using RGC innervation patterns and anatomical locations. And so here you can see these Cre-expressing cells expressing HM3D, which means that they'll have increased activity when I inject CNO. And so in control mice, we see that without any treatment, there's very little regeneration when the NOT is outlined in white. But when I increase activity within these Cre-expressing neurons in the NOT, I see a robust increase in regeneration. And we were really surprised by these results because even increasing activity in about 150 to 200 neurons was sufficient to increase regeneration compared to controls. And these regenerating axons extend up to 1 millimeter past the lesion site within 2 weeks. What was even more surprising to us is that increasing activity in one visual target in the NOT was sufficient to promote regeneration in neighboring targets as well. So we see regenerating axons innervating the OPN in the NOT, which is where I increase activity, but also there are regenerating axons innervating the NDPPN and the major targets like the DLGN, as well as the superior colliculus. And so this tells us that selective stimulation of one visual target was sufficient to promote regenerating axons to neighboring targets as well. Now, we wanted to confirm, now this is a little bit of getting into semantics, but for those of you who are interested in regeneration, we wanted to understand if these injured axons are truly regrowing and so they're truly regenerating processes, or if there are spared axons that remain connected and are now promoting collaterals and promoting, and they are the ones that are sprouting collaterals and promoting regeneration and rescue. And so we used a retrograde approach where we injected rabies, the rabies virus with a GFP tag into the NOT. And so the premise is that this would label the cells that are connected to these axons. And if we look at the retina, we should see an X number of cells that are labeled with this rabies virus. And so we looked at these controlled injured retinas, and we saw some cells labeled in the retina, as you would expect. But when we looked at the retinas from the activity mice, we noticed many more RGCs labeled in the retina. And there's approximately a 3-fold increase in the number of cells labeled in the activity group, which tells us there are presumably some spared axons that are sprouting collaterals, but that there are also injured axons that are truly regenerating. So this tells us that retinorecipient target activity is sufficient to promote regeneration of RGC axons. So to summarize, I've shown you that a distal injury model is a really good way to assess how to get regenerating axons to reconnect with their targets and really understand what's going on with the postsynaptic neurons after injury. And I've shown you that you can injure the distal, the optic tract closer to the targets in this distal injury model, and that without any treatment, there really isn't very much regeneration going on. But when you increase activity distal to the lesion site, even broadly, you see you can now promote a robust regenerative response in these RGC axons. So this speaks to the role of neural activity in brain repair and gets us wondering whether we want to at least maintain the level of activity or perhaps increase the level of activity post-injury in order to promote repair. And I've also shown you that increasing activity in specific postsynaptic neurons that receive visual input is sufficient to promote regeneration of RGC axons. And so this speaks to the role of postsynaptic neurons in circuit repair. And what's really exciting to me is that this strategy can be applicable to many other blinding diseases and optic neuropathies like glaucoma, optic atrophy, Leber's hereditary optic neuropathy, and other conditions. And even though these conditions vary in their origin, the pathology is the same in some sense, in where that RGC axons degenerating and disconnecting from those postsynaptic targets is what leads to vision loss. And so if we can increase activity and leverage these postsynaptic neurons, we can perhaps retain some of these connections and promote repair in all of these blinding diseases. What's even more exciting is that this strategy of tapping into postsynaptic neurons can be broadly applicable to other CNS injuries and diseases like traumatic brain injuries, spinal cord injuries, stroke, and even epilepsy. Especially nowadays where non-invasive brain stimulation has become more common, you can imagine that tapping into postsynaptic neurons in a circuit could be applied to promote repair. With that, I'd like to thank everyone in the Huberman Lab, especially Andrew, for giving me free reign on this project, and our collaborators in the Kolodkin Lab at Johns Hopkins and Gendron's Lab at UCSF. And a special shout out to the people who've contributed to the work, Onkar Dande, Feng Li, and Fei Wang. And I'd also like to thank my many mentors and our funding sources, and especially big thank you to Strytech for running this journal club and giving me this opportunity. With that, I'll take any questions.
Thank you, Supraja. Thanks a lot. We actually do have a couple of questions already, and so I'll dive right into it if you don't mind. So let's start with the first one. Could you elaborate on the uniqueness of the distal injury model, and how could it use to study other diseases or conditions?
Sure. So the distal injury that I've used here is a little bit different in that it severs the optic tract in the brain. So it not only is it's a unique injury to the visual system, but it also mimics certain aspects of other brain injuries, so to speak. And especially for the visual system, it gives us a way in which we're now shifting the lesion site further down, closer to the targets. And by doing that, we're promoting cell survival for a longer period. Because what happens in some optic nerve crush injuries is that even though that's a fantastic model to study specific questions, if we want to study how to promote reconnectivity and promote connection with the postsynaptic targets, then we don't have very many neurons surviving after the optic nerve crush. And so if those cells are dead and they're gone, then there's nothing left to work with. And so this distal injury sort of shifts that lesion site and gives us a better chance to study what's going on while these axons are degenerating and perhaps cell death is oncoming.
Mm-hmm. Okay, thank you. Have you looked at how individual RGC subtypes or types are affected by the distal injury?
I have not yet. So right now in this study, we've mainly looked at all RGCs, but that's definitely one of the questions that we're already diving into, and especially there are so many beautiful studies, single cell sequencing data and other studies that have shown that there's specific resilience and susceptibility based on RGC subtypes. But we don't have a good sense of how that is, if that changes in the distal injury model, and that if there are resilient or susceptible subtypes, but that's something that we're diving into next.
Maybe related to that, the spared axons or the spared RGZs. Why are they spared? Or what do you do in your injury? Maybe we'll start with that.
Yeah, so I severed the optic tract. One of the reasons that it's a partial injury is because I was really, I come from a developmental neurobiology background and I'm an axon fanatic, but in development, you see that there are pioneering axons that really promote and provide cues for the following axons. And beautiful studies from the Mason Lab has shown that if you these RGCs also sort within the optic tract, and so there are many axon-axon fasciculation cues going on. And so one of the things that I wanted to understand was if there are some spared axons, how do they promote regeneration? Are they providing any physical pathway or signals or molecular cues that can guide these regenerating axons? And the other reason was because we don't usually typically see a clinical injury where the entire optic tract is injured and all the axons die back to one lesion. You typically see that some axons are, in spinal cord, especially spinal cord injuries, you see that some axons are injured, some are severed, and some remain connected, right? And so over here, in this optic tract in the region that I'm performing my surgery, it's injury. It's particularly tricky to sever the entire optic tract without damaging the targets. It can be done perhaps at another site, but for the question that I was asking, I kind of wanted a partial injury so I can use those paired axons.
Mm-hmm. And so you're seeing that they're spared because this green tracer staining remains. If all RGCs would die today, how long would it take for the green tracer signal to disappear?
Yeah, no, that's a great question. I think so. I'm concluding that there are spared axons because when I inject my post-injury tracer, I see axons that are green as well as red. And so I know that they are still connected to the retina to receive that post-injury tracer. But there are definitely axons that are severed and they are only green. So they're disconnected from the retina and don't take up that post-injury tracer. Um, Now, to the second part of your question, I think some studies have shown that it takes approximately about 90 days for degenerating stubs to be cleared in the central nervous system. And so that's what I would expect. I think the disconnected green stubs would disappear within a 90-day period, would be my guess, but I don't, yeah.
Okay, then the question of if increasing activity in the postsynaptic neurons would promote regeneration in an optic nerve crush model as well, if you're early enough in that treatment.
Yeah, no, that's a very relevant question, and I think it is possible. I think we know that within the first week, well, a lot of the cell RGCs start dying. I think the axons degenerate well before that, after an optic nerve crush, but it is possible that increasing activity in these postsynaptic targets immediately after an optic nerve crush could promote regeneration, maybe even cell survival. But I haven't looked at that in an optic nerve crush model. But it's definitely important to see how postsynaptic target-derived signals of postsynaptic activity applies to other injury models.
Okay. And then a question, if you have looked at pattern ERG as a functional readout.
Right. No, I have not done that. I haven't looked at function ERGs in the retinas yet. Oh, but would be happy to collaborate with someone and do that.
Okay. How long would it take to fully restore RGC function approximately?
I think I'm guessing fully restore RGC function means getting them reconnected and restoring complete function. And if that's the case, I think it really, I'm speculating here, but I think that really depends on, again, the type of injury as well as if you're looking at subtypes that are resilient or susceptible. From what we know, I'm looking at a 2-week time point in my studies, and we do see a rescue of function, but we haven't looked at other sort of more refined specific visual functions to see how well those are restored. And I think other studies have shown that if you use a combination of cocktails such as the P10 and knocking down P10, CNTF, and SOX3, and all of those, then at about 16 weeks or so, you do rescue function. As far as full function, I think that's sort of the $1 million question in the field that we all need to work together to solve.
Okay, so in your last part of the talk, you showed that you have very local reactivation activation of the target regions, that you get re-innervation of neighboring regions as well. So that would suggest that the activity there in the postsynaptic target is not required for the reconnection, but rather that the activity sets up some sort of environment that promotes regeneration rather than reconnection. Isn't that right?
I think it's possible that both are happening, and that would be my guess. I think because of the degree of regeneration that I'm seeing in the short amount of time, my guess would be that stimulating a specific target maybe promotes some environmental signals that generally, like maybe like a diffuse trophic factor that generally promotes growth, but there may be specific signals in those activated neurons that are also directing. So a thing that I didn't show today was the NOT, which is where I'm increasing activity, does actually receive the highest amount of regenerating axons compared to the other neighboring targets, if you normalize for the size of the target. So it's unclear as yet, and that's one of the things that I'm going to parse in my own lab. But my guess would be that there's both taking place.
Okay. Can you identify if the regeneration you observe is coming from spared axons that sprout collaterals, or are they from injured axons?
Yeah, I think so. The last slide that I showed, if I can go back. Yeah, so that's sort of what we were really interested in that question too, and so that's sort of what we're getting to here, where if you inject a rabies virus or retrograde virus into the NOT, where we're stimulating, that virus is basically going to label any axons that are connected to the center, and so that would include some spared axons. It would include or some spared axons, but it would also label regenerating axons. And the premise is that if in control animals, what you see, if assuming they are spared axons, sprouting collaterals, or just spared axons connected to that target, you would see an X number of cells. But if there are regenerating axons, then that number would increase in the activity group. And in fact, that's what we see. We see a threefold increase in the activity group. And so that indicates that there are obviously spared axons that maybe remain connected and sprout collaterals, which would account for this portion. But there are truly injured axons that are also regenerating because of the difference that we're seeing between the two groups.
Next question. It says, since all injuries are not the same and inflammation plays a major role in the repair process, which way the immune system may be playing, or what role the immune system may be playing in particular in disease? What's your suggestion there?
I mean, we know from some really beautiful studies that there are many important immune factors that tie into the degeneration and regeneration process, which are also interconnected. So it would be really interesting to look at in this injury or in leveraging these postsynaptic neuronal activity, what happens with the immune system or immune factors and how that ties into regeneration. It's not something that we've looked at yet, but it would be very interesting to look at it. I think my guess is that, or I would want to look at both just in the distal injury context and also increasing activity, because increasing activity alone can influence microglia, which could then play a role in degeneration and how that's being cleared, which could then tie into regeneration. So I think there's a lot of interesting, potentially a lot of interesting things going on in that realm connected to the immune system, which would be interesting to parse, but we haven't looked at it yet.
Then I have a question. The distal injury model, what would be the mostly related normal injury that a patient could have that's mostly related to your distal injury model? Would that be a localized stroke, or what would that be?
Yeah, I think I'm not a clinician, but I think a localized stroke would probably be a good example. Maybe even a traumatic brain injury. That because that entire optic tract and this entire region is subcortical. So you could imagine that if there is a brain injury anywhere in the hippocampus or something above it in the cortical regions, it could impact these subcortical regions as well, potentially. So those would be my guesses, that the closest examples to the distal injury, because once that subcortical region is impacted in any way, either by a stroke or an actual injury, those axons are going to degenerate, but not in quite the same fashion as something happening in the eye, for example.
Okay, then I think we're already through the questions. Again, thank you very much for being here and presenting this beautiful study. Thank you to the audience for participating and for the good questions and discussion we had. And so I hope I'll see you for our next journal club. Goodbye.
Thank you.